Preparation to phylogenetic analysis. The gene that was used in alignment was cytochrome oxidase subunit CO1 gene, a mitochondrial gene. The accession numbers given in gene bank can be used to obtain related articles that are published. And then create a database in Excel for our own reference and obtain gene sequences from gene bank using accession numbers. Save them in the notepad in the format of FASTA and then combine with other sequences in BioEdit. Therefore, we can run an alignment again and the sequences will be ready for further phylogenetic studies. As you can see here, these are the accession numbers. So let's go in one of it. Okay, this is the article. The author, the title is there, direct submission. The entire gene sequence. So here it states submitted and direct submission. We have to look for a paper that has been published in order for us to use their gene sequences and also to obtain other gene sequences from that article. Like for example, this is another article. This is another accession number and the article here is stated unpublished. So for example, if this is the genus that we are interested in, we can actually look for articles based on this genus in Scopus or even Google Scholar. This is an example of article. Of course, it is from a different taxa, but it is also related to phylogenetic studies. Example of articles will look something like this with accession numbers, gene bank accession number. This article has been using two different genes. Therefore, there's an accession number for both the genes. So we can take as many as accession numbers as possible to conduct a phylogenetic study. So in case we are doing a phylogenetic study with more than 10 gene sequence, the best way for us to organize our gene sequences are by using Excel, whereby we can write down the taxa, the gene accession number, and the author or the publication of the article. So this will make will create a database for our phylogenetic study. So now after getting our accession numbers, what we have to do is go to Google and search for gene bank. The first option here, gene bank overview, this is what we're going to use, and this is how it looks like. And here at the search here, this is where we're going to paste our gene bank accession numbers. So we can actually paste more than one accession number here. Just insert the comma in between and then click on the search button. So there are 4 items here. We can even do more than 10, as many as we want. And in the summary here, we have to click on the FASTA text so that we can obtain all the gene sequences, and then just select all. And paste it in a notepad. Okay. So there's 4 here, and then save it. is genebank.fas. Alright. So these are the sequence that we have downloaded from the gene bank just now. What we can do is copy paste all the sequences into the alignment editor that we had just now with the same gene. So this is our aligned gene, right? So all we have to do is just copy paste all the other 4 genes into this and align it again, and that's all. We are ready for further phylogenetic studies. For the final part, an additional genetic marker, which is a nuclear protein coding gene, the NPC genetic markers. These are actually employed in studies of Coleoptera, since Coleoptera is the most diverse insect order. Compared with the nuclear ribosomal DNA and the mitochondrial genes, NPC genes are more informative and less biased in base composition, and they have been used to resolve many problematic relationships in beetles. All the steps above are repeated for this sequence of gene as well. The end result will be producing 2 different phylogenetic analyses of 2 genes, which can be used as a comparison.